My name's Susan Hillier and I'm the Dean for Research at UniSA in the Health Sciences, Allied Health and Health Science area. I take this moment to acknowledge that certainly those of us who are talking with you today are meeting on the lands of the Kaurna people, and that we pay our respects to elders, past, present and emerging, and acknowledge the ongoing relationship with country that the Kaurna people have, and proffer our support for their ongoing connection. It gives me a great deal of pleasure to introduce two colleagues from the Centre for Cancer Biology who live in the very fancy building on North Terrace, now known as the Bradley Building. And they have been working with UniSA now for a number of years. So firstly, if I introduce Professor Michael Samuel, and he is a research professor in matrix biology and heads up the laboratory for tumour microenvironments at CCB. And they're particularly looking at the microenvironment and the way that current cancer therapies, which look at the tumour cells, but maybe that they can have different approaches around different aspects of that environment. Michael has been an ARC Future Fellow. He's won Emerging Leadership Awards from the ANZSCTD. You can ask him what that stands for. And he's now been working with CCB for 10 years. So Michael, I'll hand over to you. Thank you.
So thank you very much for this opportunity to present at the Adelaide Biomed City Research Minireview. I particularly want to thank John and Tamara for the invitation and for organizing this. So I'm going to talk a little bit about the work that we've been doing in figuring out how tumors make a permissive microenvironment, and show you some of the tools and the models that we use that may be of interest to people in Adelaide here. So just a bit of an overview about the cancer microenvironment. So tumors don't exist in a vacuum. They're in fact surrounded by a whole host of different structures and features. Most of these are essentially a co-opting of normal parts of the tissue. One of the things that we're interested in is the extracellular matrix, which consists of polymeric proteins, some of which are remodeled, as you can see here, in the context of cancer, to provide mechanical cues that are important for cancers to grow. There are also blood vessels that are co-opted to provide nutrients for the growing cancer. And another cell type called fibroblasts that my lab works on that have probably been underappreciated for a while now, that are shapeshifters. They're able to do a lot of different things in the context of the microenvironment, and tumors are usually very good at co-opting these cell types for their own purposes. There are also a whole host of immune cells and other cell types that a lot of people have been working on in the field that are often used by the cancer during a disease progression. So the microenvironment can exist in two different forms. One of them would be the tumor suppressive microenvironment, and under these conditions, these normal cell types, including immune cells and fibroblasts, suppress tumor growth by either secreting factors that inhibit growth of the tumor or by actively killing tumor cells. But it can also exist in a tumor promoting form. And under these conditions, you can see all of these red arrows here, which indicate communication between the cancer and these cells within the microenvironment. The cancer changes the properties of these cells such that they then produce factors that you can see here indicated by the blue arrows that promote disease progression. And it's this process that we are particularly interested in. And you will see that my favorite cell type, the fibroblast, is missing from this panel here. So I've just taken a panel from a recent review from the lab to demonstrate how this works. So tumors are capable of producing a host of different growth factors, and this list is growing. And some of these factors act on fibroblasts to modify their behavior. So fibroblasts in this context are genetically normal, but they're acting under the influence of the tumor and are therefore working against the interests of the host. So fibroblasts can be converted into pro-tumor cancer-associated fibroblasts. They can become senescent and secrete senescence-associated factors that promote tumor progression. They're also able to produce and remodel the extracellular matrix that provide mechanical cues that compress or pull on the tumor, and as a result, they induce mitogenic growth. They're also able to hijack other cell types within the microenvironment. So these pro-tumor CAFs then secrete factors that recruit some immune cell types that are tumor promoting. So we are particularly interested in a group of tumors that are mechanoresponsive, and those are tumors whose biology is strongly influenced by mechanical force. And the reason these tumors are influenced by mechanical force is because they're capable of interconverting mechanical force into biochemical signaling. And a few examples of signaling pathways are listed here, and you may recognize some of these as very classical biochemical signaling pathways. And this interconversion is called mechanotransduction signaling, and it's of increasing interest in the cancer biology field. A series of preclinical models are used by my lab in order to study how mechanotransduction signaling is brought about and how it is used to hijack the tumor microenvironment. One of the models we use is this two-stage chemical carcinogenesis model of cutaneous squamous cell carcinoma, which involves the initial application of a carcinogen to the skin, followed by repeated application of a tumor-promoting agent. And this allows us to very nicely study the extracellular matrix, because the tumors that are produced are very rich in extracellular matrix proteins. We're also using a colitis-associated model of intestinal carcinoma, which very similar to this two-stage carcinogenesis protocol involves the administration of an carcinogen that's very specific to the intestine of these mice, and repeated promotion of tumor growth by the administration of dextran sulfate, which induces inflammation in the gut. And we use this model to look at tumor growth in vivo, but also to generate organoids to see how various signaling pathways, including our favorite signaling pathway, the Roadrock pathway, regulates tumor growth via the microenvironment in this model. And also Sarah Boyle in the lab brought this new model to us together with a very long-term collaborator, Marina Kochetkova. And this model is a breast cancer model that involves the driving of tumor growth in the mouse mammary gland by the polyoma middle T antigen. And this allows us to do a few very nice things, which includes the production of tumors that then can be manipulated using siRNA or shRNA, and then engrafting those back into wild-type FVB immune-competent mice, or allows us to actually ex vivo compress or pull on tumors to induce the kind of mechanical influences that I was talking about. And then we can analyze the tumor by immunofluorescence and various other processes as they grow within the mouse. So our lab is interested in the Roadrock signaling pathway. This pathway is well known to regulate actin-myosin contractility by regulating both actin polymerization and myosin contractility. But a few years ago, we demonstrated that this pathway is also involved in regulating the extracellular matrix and increasing its mechanical stiffness. And this is associated with progressive disease, but also can be co-opted to enhance wound healing. And so we published on this a few years ago. Now, the Roadrock signaling pathway is activated in all three of the models that I showed you before, but I'm showing you the data here in the breast cancer model. And also particularly here in human breast cancers, you can see that as tumors become invasive, they overexpress this protein ROCK, both isoforms ROCK1 and ROCK2, and also hyperactivate ROCK. And you can see this by immunohistochemical analysis of one of the substrates of ROCK kinase, myosin phosphatase, which is higher in the phosphorylated version of which is higher in invasive ductal carcinoma compared to normal. Now, over the years, since its establishment as an important regulator of tumor progression by my mentor, Mike Olson, at the Pitzen Institute in Glasgow, when they demonstrated that membrane blebbing associated with apoptosis is mediated by ROCK. This membrane blebbing is also important in a particular mode of cell motility called amoeboid movement, and it was initially thought that this was the main function of the Roadrock signaling pathway in tumor progression. However, we demonstrated that when you activate ROCK in the context of cutaneous squamous cell carcinoma in mice, the tumor promotion was dependent on the production of a very stiff extracellular matrix by these tumor cells. And we didn't work out the precise mechanism by which this happened, but this was the first time it was demonstrated that the Roadrock signaling pathway regulated the tumor microenvironment. We then went on to demonstrate one of the mechanisms by which the Roadrock pathway is hyperactivated in cutaneous squamous cell carcinoma. And also, as I said before, worked out a way in which this could be exploited to enhance skin wound healing. And I'm happy to discuss this later if necessary. The Olsen lab was also able to demonstrate that the Roadrock signaling pathway in pancreatic cancer promoted tumor growth by a mechanism involving the breakdown of collagen polymer within the microenvironment of pancreatic cancer. So there's this kind of dual mechanism by which this pathway works. Paul Timpson, a collaborator at the Garvan Institute in Sydney, then used the fundamental discoveries that the field had made over the last few years to very elegantly demonstrate that this could be exploited in therapy. And using preclinical models, his group demonstrated that if you transiently inhibit the Roadrock signaling pathway prior to administering chemotherapy in a few different models, you could actually enhance the efficacy of this chemotherapy. And they showed that this was by weakening the stiffness within the extracellular matrix in this process. So that was quite an interesting translational aspect to this work. More recently, we demonstrated that the way in which the Roadrock signaling pathway regulates the extracellular matrix and establishes a tumor-promoting microenvironment is by hijacking an unfolded protein response signaling arm to produce this protein called CREL2. And I'll tell you a little bit more about this now. This is a little vignette of some of the published work. So in the mouse mammary cancer model, when we activate the Roadrock signaling pathway by genetic means, these tumors develop a very stiff and highly collagenous extracellular matrix, which also contains other tumor-promoting proteins like tenascin C. And this is associated with the recruitment of fibroblasts into this extracellular matrix. Here we've marked it using this particular marker of fibroblasts, which is the beta isoform of the PDGF receptor. Now, and this results in rapid growth of the tumor. So you can see that here. So this is the kinase dead tumors in blue and the ROCK activated tumors in red. But this is not actually the tumors themselves, but the fibroblasts associated with those tumors that appear to be tumor promoting when you just take them out of these mice and co-engraft them into another mouse with tumor cells that don't have ROCK activated. And we've worked out that the mechanism by which tumor cells hijack these fibroblasts is by producing this protein called CREL2, which is enriched in the secretome of tumor cells in which ROCK is activated. And when we knock down CREL2 in these tumor cells, we can abolish the ability of these tumors to hijack these fibroblasts. And the fibroblasts produced by tumors in which ROCK is activated, but CREL2 is knocked down, can no longer enhance tumor growth. So this established a novel signaling axis between ROCK and CREL2 that we could potentially, will be informative in how tumors hijack their microenvironment, but also could potentially be targeted as a way in which we could normalize the tumor microenvironment to stop tumor growth. And this is the data associated with the mechanism by which ROCK activation produces CREL2, and that is via hijacking of one of the arms of the unfolded protein response, mediated by this PERK protein. There are 3 separate arms of the unfolded protein response. It's only the PERK arm that's hijacked by ROCK activation. And when you knock down PERK, you can abolish the ability of tumors in which tumor cells in which ROCK is activated to produce CREL2 and elicit particular phenotypes in fibroblasts. And in this case, we're looking at a phenotype which is the enhanced ability of fibroblasts to move in 3 dimensions, which we measure by inducing them to move in 3 dimensions towards a chemotactic gradient or along a chemotactic gradient in 3 dimensions. So this led us to have a look at whether CREL2 is in fact associated with breast cancer in human patients. And we looked at a data set of 2,000 samples of breast cancer, invasive human breast cancer, to demonstrate that a high level of expression of CREL2 in tumors is associated with a reduced 5-year survival rate in these patients. This suggests that the mechanism that we've shown in our animal models is in fact relevant to human breast cancers. And in fact, interestingly, high levels of CREL2 are associated with invasive ductal carcinoma compared to all other types, but also associated with the triple negative receptor status compared to all other receptor statuses. And as many of you may know, the triple negative breast cancer subtype is one of the least treatable and most likely to invade and metastasize in women. So this suggests that the phenotype associated with some of these more aggressive cancers is associated with CREL2 production, and validates this as a potential axis that should be targeted. So where to from here? I've shown you, well, what we think is that CREL2 is in fact a prototype. It's a prototypic of a suite of differentially secreted molecules downstream of this Roadrock signaling pathway. We've also identified a few other molecules that are of interest, and I'll tell you about one of these in the next slide. And this suite of proteins.
Michael, we probably need to wind up now. Just
okay.
Yeah.
All right, so anyway, I won't tell you about the new data then. I will just move on to thank the people that did this work, a long-term collaborator, Marina Kochetkova, Sarah Ball in the lab, and Zahid, whose data I also showed you today. Our collaborators at the Walter and Eliza Hall Institute, the Garvan Institute, and also the Center for Cancer Biology. And these are our funding bodies.
Thank you very much, Michael. That's fantastic work. Look, in interests of time, I'll just encourage participants to get in touch with you with questions.
Yep.
And we'll move on to our next speaker, who I'm delighted to introduce, Professor Stuart Pitson. Professor Pitzen is also in the Centre for Cancer Biology in the Bradley Building at UniSA, a collaboration between SA Pathology and UniSA. Stuart is an NHMRC Senior Fellow, and he's the chair funded by the Neurosurgery Research Foundation. His work is in lipid metabolism, particularly looking at fundamental pathways, and of particular note, taking his research into the commercialisation space, which I know is of great interest to people, and some of his adventures there hopefully will form some of his story today. So over to you, Stuart. Thank you.
Thank you, Susan. Right, so I will give you a bit of a whirlwind tour of our research and try and give you some indications of where we can potentially collaborate with multiple people across the state and potentially value add to research. So really the overview of my talk is really looking at our fundamental research in sphingolipid metabolism and how we managed to translate that towards the clinic. Then I'll talk really the main title of my talk has been in patient-derived xenograft models for cancer research. So we have multiple angles there. I want to talk about today is our work in the brain tumor space with glioblastoma, but then also with acute myeloid leukemia and some of the advanced models that we're operating with there. So with sphingolipid metabolism, this is, don't get scared by these pathways. I used to hate these pathways in undergrad biochemistry, but essentially this is sphingolipid metabolism where you take palmitic acid and serine, so a very common saturated fatty acid and an amino acid. They get condensed into a series of different lipids, which then go down to some of the key lipids, which are ceramide, which has been long known to be involved in inducing cell death, It also is a pro-inflammatory molecule and can induce fibrotic effects. Further down the pathway, we have sphingosine-1 phosphate, which has an opposite effect in many respects to ceramide in inducing cell proliferation and cell survival. There's also a key player in immune cell trafficking, particularly T cells, and there are drugs in the clinic at the moment targeting sphingosine phosphate receptors in the immune system to suppress that system. Sphingosine phosphate is also involved in hypertension. So we've been involved in really fundamental research in understanding various different enzymes in this pathway. Sphingosine kinase, which I've highlighted in red here as being a key one, but also dihydroceramide desaturase and a number of other enzymes that I will touch on today. Again, most of that's been fundamental research, but we've been particularly pleasing to be able to take, as Susan said, some of this through commercialization towards the clinic. So the first bit I'll really talk about, or the only bit I'll talk about this pathway today is this conversion of dihydroceramide to ceramide. So on my next slide, this is essentially the same step that I just mentioned there. So dihydroceramides get converted to ceramides by this enzyme called dihydroceramide desaturase 1, DES1. Incredibly small difference between these lipids. Basically you're introducing a double bond here to generate these ceramides. You might think, well, big deal, what's that going to do? Actually changes the properties of these lipids quite substantially, and there's a whole slew of studies that have looked at the binding partners of these lipids, and they're quite different between dihydroceramides and ceramides. Membrane properties are quite different. These lipids all tend to live within membranes because they have these two fatty acid chains, and the properties of those membranes where you have high levels of ceramide are completely different to those where you've got high levels of dihydroceramide. As I said before, ceramides tend to be inflammatory, fibrotic, and they can also induce insulin resistance. But on the flip side, these dihydroceramides, because they have different properties, tend to amazingly have different, quite opposite effects in anti-inflammatory and anti-fibrotic. So this has led us to examine the inhibition of this DES1 enzyme as a potential therapy for fibrotic diseases. So, we're looking to develop inhibitors of dihydroceramide desaturase 1. And through that, we were fortunate to interact with the Medical Research Commercialization Fund. So myself and my collaborator, Bernie Flynn from Monash Institute of Pharmaceutical Sciences in Melbourne, we co-founded this company, Sincera Therapeutics, through a $7 million investment from the Medical Research Commercialization Fund. So with this, Bernie is a medicinal chemist, I handle the biology, and we've been able to generate, can't show you too much data at the moment because it's commercially sensitive, but we've been able to generate very potent DES1 inhibitors that show very good anti-fibrotic activity in a whole range of different mouse models of fibrosis. So we are very much hoping to be able to push this work towards the clinic in the next year or so, into clinical trials in the next year or so. So I'm very keen on chatting with anybody who wants to know about this commercialization path that we've had, particularly in terms of our interaction with the MRCF. And I'm very happy to chat with anybody who wants to explore that further. Now, if I change gears again to go to some of our patient-derived xenograft models. Really, the first one I want to talk about is glioblastoma. We've invested a lot in the last 4 or 5 years in being able to set up a lot of good models of glioblastoma. So glioblastoma is a pretty poorly treated disease at the moment. The median survival is 15 months and it really hasn't changed for 30 years. And the best drug on the market is temozolomide, which only enhances the median survival by 3 months. So it goes from 12 months to 15 months with temozolomide and radiation therapy. So there's an enormous need for new therapies for glioblastoma. And so that's what our motivation has been, is to try and set up some good patient-derived xenograft models. So we've been very much aided by the clinicians of South Australia here, so particularly Santosh Pranus at Flinders and the South Australian Neurological Tumour Bank have been pivotal in being able to gain access to a lot of patient material. So from that, we take that material and we do tumour histology and take bloods to look for biomarker discovery. A lot of that's done with collaborations both here in the CCB and broader. We also, one of our main purposes is to generate glioblastoma cell lines, so low passage cell lines. These are grown as gliomoneural stem cells. So they're essentially the tumor initiating cells, and they can be passaged for quite a long time and still maintain those patient features, patient cell features. With interactions with Guillermo Gomez here at the Center for Cancer Biology, he has got a lot of glioblastoma explant expertise happening. So where you can basically take the solid tumor, dissect it into small chunks and grow those in the laboratory. They'll expand in the laboratory. That very much keeps the tumor microenvironment intact. And so it should be a much better way of drug screening platform. He also has, from our glioblastoma cell lines, managed to generate glioblastoma organoids, so a three-dimensional culture of cells. Again, more representative than two-dimensional cultures, and that allows of what the tumor is like, and that allows for studies to enable studies to look at tumor invasion and normal tissue interactions of the tumor cells. One of the big things that we've invested in, though, is really generating these intracranial glioblastoma xenographs. So where we can generate xenographs and which have an intact blood-brain barrier, and then we can apply drugs to these models, which are really the key preclinical step in the process. Throughout many of these processes, we do genomic analysis. Surprisingly, the glioblastoma patients' tumours don't really get analysed very much at the moment for genomics, because most patients, nearly every patient, will receive a single drug, which is temozolomide. There's no sort of personalised medicine in this space, mainly because of the blood-brain barrier preventing most drugs getting in. So temozolomide does get into the brain, so that's why it's used, but it's not a particularly good drug. So we do a genomic analysis as well to be able to help stratify for anything we find in drug therapies to be able to then push this towards personalised medicine. So for all of this, we look at these models for evaluating the efficacy of new drugs, both we're generating and also those that are being generated by collaborators, and screen these drugs alone and in combination, both in the cell lines and organoids, explants, and in the xenographs. And also we want to understand fundamental biology of glioblastoma because that will then help advance new drug therapies. So at the moment we have generated 35 glioblastoma cell lines from 35 patients. These all encompass the 3 known molecular subtypes of glioblastoma. As I said, the molecular subtyping is not done for really therapeutic purposes, but there are clearly three molecular subtypes of glioblastoma. And so our tumour samples so far have encompassed all of those. And we have multiple explants with Guillermo Gomez and organoids and multiple xenographs as well. So this is just some of the examples of some of the work that we've got. So here are some intracranial xenographs. We have been fortunate to get funding from the Neurosurgical Research Foundation to set up intracranial xenographs, so where we can inject these cells straight into the brains of mice, and these are all, and we've managed to develop systems where we can transfect or transduce the cells with luciferase containing vectors and then image the mice during their tumour's progression. These first 2 mice are using cell lines, well-established cell lines, so U251 and U87, which are 2 of the most well-used cell lines in the field. But then here are the other 3 are 3 of our patient-derived xenografts. So again, all growing in the brains of the mice. We can look at taking the mice out to their ethical endpoints. We normally see neurological defects in the mice, and that's when they're taken down. And you can see here, that's a U87, a very aggressive cell line. The other cell line is a U251, which is in the red line. But then our 3 patient-derived cells tend to fit within this scheme. So again, they go down quite uniformly. So this allows for quite good therapeutic interventions because we can use a smaller number of mice because all the mice tend to go down very, very similar point in time. That's the beauty of using a stereotactic setup, so we can implant the tumour cells in exactly the same region of each mouse, and the tumour will grow in a very comparable manner and generate neurological defects in a very comparable manner. And so because they're patient cells, some will kill the mice or will get to the endpoints faster than others. And so we have at least a number that can be used in therapy interventions. What I want to show you though is that U251, so this is a cell line that's been around for many years. You see these do not grow at all like a patient tumor. So they grow as very much a very defined normal brain tumor mass. So there's a very clear demarcation between the normal brain and the tumor, whereas with our patient-derived cells, we actually have this very diffuse tumor margin, which is much more like what you would see in a patient. So normal tumor cells invading into normal surrounding brain. So these are a very, very good model to be able to study, much better than these cell lines, which we would not recommend anybody using. The last thing, I think Susan's come back online, so I'll wrap up fairly quickly. We have models of acute myeloid leukemia as well, which Jason Powell has been really championing here. We have a lot of patient material that's been assisted by Sakharov and David Ross, and we've also stored a lot of the samples through that Sakharov process. We have 13 different acute myeloid leukemia patient samples that we can engraft into mice. These are all primary cells straight from the patient, frozen down, which we can go and engraft into the mice. Nothing has been passaged through the mouse, so many other groups will engraft cells into mice, expand those up in the mice, and then harvest those cells to do re-engraftment. We think that skews we know that skews the clonal architecture of the AML, which is a very heterogeneous clonal disease. So we prefer to go back to the original patient samples, which we can do because we've frozen down many ampules from leukapheresis products. So we have 13 that engraft reproducibly in mice. These cover a whole range of different AML mutational statuses. And so we can then start looking at personalized medicine approaches for these, and with our own drugs, but we've also a series of publications with collaborations with other people using standard PDX models, which are essentially engrafting these cells by tail vein injection into NSG or non-skid mice, may or may not irradiate depending on the patient sample. Those cells then engraft into the bone marrow of the mice, reproduce a human AML, which we monitor through the human CD45 in peripheral blood, and then we can start therapy and establish disease and analyze the human AML burden in the bone marrow and look at survival analysis. I do want to just share, so this is just a little bit of data where we've published in essentially where we do get a reduction in leukemic burden targeting one of our lipid enzymes, sphingosine kinase 1. Here we see, this is brown is the human mitochondrial staining. So we have in the vehicle control cells, we have huge number of human AML cells here, and in the drug-treated mice, we see the bone marrow has a lot more, much less brown, so much more leukemic burden, but also normal mouse hematopoiesis is coming back. So we're not killing the normal hematopoietic system, and so this is very exciting data. We get survival of the mice extended. I will wrap up very quickly. So we also have another more advanced model of this. One of the problems with these simple PDX models for acute myeloid leukemia is you only tend to select for a single AML clone. There are multiple clones in pretty much every AML patient. So we've generated, with the help of Dan Thomas and particularly championed by Jason Powell here, we've generated this humanized bone marrow ossicle PDX model where we can take mesenchymal stem cells from the bone marrow of healthy donors, basically take those cells and engraft them into the flanks of mice. They then grow as bone organoids, human bone organoids in the flanks of the mice subcutaneously. They exhibit all the facets of normal human bone marrow, including bone and stroma, and they have mouse hematopoietic cells invading into them. And then we can take the AML cells, inject into these ossicles, and that can establish disease. We're still developing the model fully, but the beauty of this is that the full clonal architecture of the primary patient material, so the various clones that are in that primary AML sample, will be recapitulated in that bone marrow of that humanized ossicle. So this provides us great opportunity to actually look at the full spectrum of AML clones, particularly those that may then drive relapse from therapy. This is just some data showing these are the small ossicles that have been growing in the mice. And this is, with the help of Paul Anderson, some of the staining of the ossicles. This is a micro CT analysis of the ossicle showing that you actually have this hollow bone marrow-like structure within this inside of the ossicle, and that there are all the features of ossicle, of bone and bone marrow engraftment, a bone marrow structure of these ossicles. So very exciting model to progress. We have lots of other research areas of interest, which which you can see there, and a number of different collaborations. The ones I've mentioned today are sort of main ones. And again, thanks to all of the group that have been really working hard to generate these models, and from particularly NHMRC, we've been particularly fortunate in getting support from the NHMRC and a number of other organisations. With that, I'll happy to take any questions.
Great. Okay, thanks. Thanks, Stuart. Thanks, Michael. Great talks. Again, I'll really encourage our participants to contact you directly, Stuart, and you directly, Michael. I'm sure you're both very approachable fellows, and they can take up some questions with you directly. So thanks everybody for participating, and we look forward to seeing you again next week.